Go ahead.
Imagine being able to choose your baby's eye color, hair color, and talents, and everything about your child before it is even born. This seems like a fictional and futuristic concept, yet we are much closer to this reality than previously imagined, due to recent developments in biomedical science, such as CRISPR. However, many scientists and ethicists alike are concerned about the widespread misuse of CRISPR technology.
CRISPR, also known as clustered regularly interspaced short palindromic repeats, allows for genes in DNA to be precisely edited, affecting the genetic code and allowing it to alter the genes of a fetus. It acts as tiny molecular scissors, removing the targeted gene and replacing it with alternative DNA. The consensus is that regulations need to be put in place to limit the misuse of gene editing technologies such as CRISPR.
This brings forth questions of what CRISPR technology means for the future. So we attack this question from three main perspectives. The first one being the question of what does CRISPR entail from a medical standpoint? Then, what are the ethical considerations of gene editing in general? And finally, what does Judaism have to say about a future with such technology?
Firstly, we consider what CRISPR entails from a medical perspective, holding many considerations regarding free access to frivolously manipulate germline DNA. As of now, CRISPR has its deficiencies, and these deficiencies can cause medical complications. It is difficult to control exactly how many cells are modified, which raises a risk of cuts being made somewhere else in the genome. These potential dangers not only affect the intended target of CRISPR, but the embryo's progeny as well, because generations upon generations will carry the same mutation. This diagram shows how hereditary genes are carried throughout generations and how they can be dangerous as well.
Since CRISPR may pose a detrimental effect on an embryo's progeny, it can perniciously impact the future of genetic diversity in humanity. Genetic diversity exists when DNA sequence of distinct individuals differs to a certain degree, which creates different traits. So, for example, eye color varies throughout the world, and one's determined eye color is dependent on the appearance and disappearance of a genetic variant called alleles. So, when one allele is expressed, it diminishes the appearance of other alleles. So, balanced allele expression is very important because the immune system relies on this genetic variability to fight off pathogens. So if an entire population of people are genetically modified to express a similarly weak allele, then any exposure to any new type of pathogen could lead to our extinction. Therefore, preserving the genetic diversity is necessary for our survival and can be done through regulating CRISPR.
Aside from the impact on genetic diversity, CRISPR clinical trials are currently being conducted due to the novelty of the technology. This chart shows how CRISPR clinical trials are moving forward and are favored by regulators, and are likely to be FDA approved in the near future. When a new therapy such as CRISPR becomes FDA approved, the expenses are usually costly. This would lead to the development of biosimilars because of the cost. Generic CRISPR therapies would be cheaper in price and possibly dangerous because some failures to the mechanisms may be overlooked, as it is created without FDA oversight. This increases the possibility of people misusing CRISPR technology to create so-called designer babies while making substantial changes to the human gene pool.
There are many ethical considerations regarding CRISPR technology and its potential outcomes. This will mainly be explored through a risk-benefit ratio, considering both the procedure itself as well as the outcomes and how it affects the individual and society as a whole. So as you can see from this chart, over half of American adults believe that using CRISPR technology for reducing the risks of diseases and conditions is an appropriate use of this technology. So now you might be wondering, what sorts of genetic diseases and conditions could CRISPR actually treat? Well, CRISPR can be used to eliminate diseases that are caused by genetic mutations, diseases such as sickle cell disease or conditions such as Down syndrome.
There could also be unintended consequences following CRISPR therapy, such as the edited embryos becoming a mosaic of cells, which is a mix of edited and unedited cells. When the cells undergo mitosis, some cells inherit unrepaired DNA, which is a big problem, as it means some cells can grow uncontrollably, possibly even leading to cancer. Putting a child in danger like this is simply considered unethical.
As the philosopher Edmund Burke states, the greater the power, the more dangerous the abuse. CRISPR is a powerful piece of technology, and with this power comes a possibility of it being misused in extreme ways, such as creating designer babies out of personal interest. Although CRISPR has many potential benefits, the risks need to be solved before widespread CRISPR accessibility. Therefore, it would be unethical to utilize CRISPR before fully developed.
Judaism considers a fetus until 40 days from conception as merely water. Considering this, what does Judaism have to say about the future of genetic engineering in embryos, knowing the great potential of CRISPR technology? So when discussing the Judaic approach to genetic engineering, the ethical concerns are always discussed. For example, Dr. Fred Rosner, a medical professor and expert in Jewish medical ethics, looks at genetic engineering and screening as permissible when used for treatment, cure, or prevention of disease. He says that such genetic manipulation is not considered to be a violation of God's natural law. Continuing from the same ethical perspective, he says that overall, Judaism does not permit the alteration or manipulation of physical traits and characteristics, when such change provides no useful benefit to mankind. In summary, Dr. Rosner believes that CRISPR technology should certainly be used when used for prevention or cure of disease, but when the overall result provides no clear benefit to mankind, such as change in hair or eye color, it is prohibited.
When discussing technology such as CRISPR, the common question is whether or not we are playing the role of God, and if so, if that is allowed. So, to answer this question, the previous chief rabbi of Great Britain, called Rabbi Emanuel Jacobovich, in his book Jewish Medical Ethics, claims that manipulating the genetic building blocks of humans through laboratory alterations is indeed considered playing God, and thus is prohibited. This is just one approach to the controversy of CRISPR technology and its potential widespread accessibility.
From a rabbinic approach, many authorities believe that if a cure for a disease is discovered, that cure would certainly be permissible for human use. Thus, it should certainly be permissible to cure or prevent these diseases before a baby is born using genetic engineering. This is a more balanced approach to the topic, which highlights the idea of taking advantage of the resources given to us by God. A current example of this is how today many vaccines are made out of natural products like eggs. This is a very similar concept to how, if God gives us the resources, scientists and biomedical engineers should be able to expand and create with these resources to enhance our lives.
Due to the clear need for regulations of CRISPR, we propose 4 viable solutions. Firstly, clinical trials should be conducted with animals to determine potential genetic mutations caused by gene editing, as shown here with a genetically modified mouse that grew an ear on its back. They should specifically be done with animals, as clinical trials can limit an embryo's viability. Funding CRISPR clinical trials would cost roughly $5 million, and this would be extracted through government medical funding. Once the technology is fully developed, access to physicians would be regulated as well.
The American Medical Association, or the AMA, should put in place stringent restrictions for physicians to abide by when providing CRISPR technology. And physicians should also control the types of genes targeted by CRISPR. The restrictions should also make sure that CRISPR is only used for medical purposes only and not for opposed to personal interest. Additionally, physicians who abuse CRISPR technology should have their medical license revoked. For instance, biophysicist He Jiankyu announced at a medical conference that he had successfully edited the cells of embryos to be HIV resistant. The embryo's germline did not contain HIV. However, it was impossible for the embryos to ever contract HIV. His medical license was revoked due to the clear misuse of this technology.
Also, physicians should refer to prospective parents wanting a designer baby, genetic counselors, sperm or egg donors, and adoption. Although these alternative options do not guarantee the ideal baby, they are more safe and viable options considering the novelty of CRISPR technology. Controversy emanates from new technologies such as CRISPR, which can be used for its great benefits in medicine. However, CRISPR can be misused to create designer babies, putting the babies at risk considering potential dangers of germline gene editing and detrimental eugenics. CRISPR needs to be further improved and regulated to eradicate genetic mutations. Therefore, it is crucial that bioengineers further developing CRISPR resolve its potential dangers and avoid throwing out the literal and proverbial baby with the bathwater.
Nathaniel, give one specific way that your thinking changed as a result of learning about Estie's findings.
Well, in my individual research, I really went into depth as over the broad overall ideas in CRISPR, and then in her perspective, being the medical perspective, she went into depth about the physical things that went into this technology and all the risks coming from the medical standpoint. And these were things that I hadn't actually researched in my own individual research, so that would be one way that my thinking changed as a result of Estie's findings through the medical perspective.
Rachel, in what way did you improve your ability to work with a group as a result of this project?
I think that we were able to, that we were all able to really improve by improving our group dynamic. Since at first, we all sort of split up and did our own individual slides, and we didn't really look at each other's until later, until after the fact. And I think that during this process, we really did improve with our inclusiveness of everyone and knowing and understanding each other's perspectives.
Esther, what is a way in which your team's resolution makes you think differently about your own individual research?
So my own individual research was about the medical perspective, and all the solutions, they all are implemented considering all of our perspectives, such as Rachel's, which was ethics, and Nathaniel's, which was Judaism. Nathaniel's focused on the Judaic ethics of it, which helped me realize that there needs to be a line drawn between CRISPR being used for medical use and for personal use, and how Judaism plays a role in that, in what they say regarding embryos' viability and all of that.
Thank you.
Thank you very much.